originally from Barcelona in Spain, but I have been happily living in UK for the last 10 years. I'm very happily in Bristol for the last 2 years. It's a fantastic city that actually reminds me a lot of Barcelona in many things, like cultural level, music, etc., etc. And also, my academic career has kept moving from one place to another. I did a degree, an undergrad on biomedical sciences with a lot of interest on genetics, but then I discovered evolution. It was a big mind blow for me. And so I did a PhD on evolution, on using genes to understand the evolution of organisms, of animals specifically. And then after that, I wanted to learn genomics because everyone was doing genomics. So I came to the UK and spent 5 years in Oxford, learning to do genomic analysis and comparative genomics. And then after that, I got a lectureship in Essex, and more recently, I got a senior lectureship here in Bristol. So I'm going to try to, when they asked me to give you a taster lecture of the kind of things we do here, I thought it was a great chance, a great opportunity to explain how can we use very basic concepts like evolution to solve very important societal problems like biomedical issues or agriculture issues. All right. So I'm going to start from the beginning, talking to you about the tree of life. So you are all familiar with this very important book. That's The Origin of Species, that was written by Darwin and published in 1859. And Darwin was British, was English. So this book is a big deal here. And one of the central concepts, and probably one of the things that most people know very well, is the idea of natural selection, no? How a mechanism that explains how a species evolve. However, sometimes I get a little bit annoyed because there is another very important concept in this book that is often overlooked. Okay, and that's what's in the title, is the origin of the species. So here you can see, for example, the first doodle of an evolutionary tree that Darwin made on one of his notebooks. This tree is not in the origin of the species, not in the book, but it was the germ of this idea. Okay, so let's talk about the origin of the species. So basically, we need to define first what is a species. There are dozens of species concepts, all right? It's one of the most philosophically debated ideas in biology. So I'm going to use the most mainstream one, the one that most people use, that is called the biological concept of a species. That says that species are groups of organisms where two individuals are capable of reproducing and produce fertile offspring, all right? Usually producing sexual reproduction. There are many problems with this definition. It only applies to organisms with sexual reproduction, so bacteria that are mostly asexual, this definition does not apply to them. Not always two members of the same species can produce fertile offspring. So you try to put together two males of the same species, they can't usually. So etc., etc. But that's the operative definition of a species that we all use. So where species come from, how new species evolve. All right, this is a process we call speciation, the genesis of new species. This is what Darwin called the mystery of mysteries. Okay, poor Darwin, he was living in a time in which there was no knowledge of genetics or developmental biology. He didn't know about Mendel, for example. It was very hard to explain some of those mechanisms. Okay, so basically we have a species. I'm going to explain you how speciation works. So we have a species that is made up of different populations. Here you have 3 populations, each of those blue blobs there. And then, as we defined before, basically species is based in the idea that genes can flow between individuals or populations, okay? Because they do this by means of reproduction. You have populations that can exchange genes, and they all belong to the same species. Now, what is going to happen is that one of those populations is going to become isolated from the others. There's some barrier that is going to stop those genes going from one population to another. The barrier might be a geographical barrier. Maybe a river emerges between this population and the others. Maybe a mountain range. Maybe some of the species get trapped in an island. Sometimes those barriers are not geographical, they are behavioral. Maybe as a population develops mating rituals that are not understood by the other population, so they don't reproduce anymore. No matter the reason, now you have that those two sets of populations are going to be isolated, one from the other. And they're going to start to accumulate their own mutations and to become different, to diverge. They diverge so much that if you give them enough time, if they get to be together again, they cannot produce fertile offspring. Okay, so then by definition now you have two new species, species B and species C. All right, so this is how speciation happens. So that means that if all the species are related by these descendant relationships, because all they have evolved from an ancestral population, we can illustrate, we can draw those relationships using a metaphor, the metaphor of a tree, of an evolutionary tree. For example, we have here two species of cats, the leopard and the domestic cat, and then we can put them together using those branches, meaning that they descend from a common ancestor. And now if we start to add more animals to this tree, then we can place them on the tree based on the relationship to the previous animals. For example, if we add a wolf, this wolf will be less related to any of those two cats than one cat to the other. So basically what you are having is that you can describe those relationships using those nodes, those branching patterns, which are going to tell us how close in evolution those animals are when they share a most recent common ancestor. So the leopard is more related to the domestic cat because it shares a more recent common ancestor with a cat than with a wolf, right? And this is what we call an evolutionary representation of the evolutionary relationships of organisms, and can be also called phylogeny, using the Greek terms for lineage and origin. All right, so basically phylogeny means the origin of the species, and we use this to classify, to check, to reconstruct the evolutionary relationships of everything. All right, so this is one example from vertebrates. No, here we have mammals with ourselves and cats, which are close related to reptiles, and reptiles and mammals together are more related to amphibians than to fish, and so on and so on. Okay. Oops, I went backwards, sorry. So that brings me to a concept that Darwin put in his book that I think is one of the most transformational concepts in biology. Okay. And basically, Darwin had this idea that there is a common pattern, a unity of life. All right. So basically, he was thinking, he suggested that all living organisms are closely related. Okay. So we all share a single last common ancestor. So this is the tree of life. So here you have all the extant live beings. So here you have animals, here you have bacteria, you have other types of bacteria here, the archaeas. Basically, if you keep traveling back in time and reconstructing all the ancestors of those organisms, at the end, all the branches collapse in a single point, okay? And this is what's called the last universal common ancestor, Luca for friends, okay? And I think this is very transformational because it's not a metaphor, it's not a poetic attempt to explain nature, it's a scientific fact. All living organisms are related. So if you go out on the street and you look to a tree, or you look to the lettuce in your salad, or you're having a pizza, we will see later, everything that you see that is alive or was alive recently is related to you. If you travel in time back enough, you will share a last common ancestor with those organisms. Okay, so that means, for example, the origin of life is thought to have happened 4,000 million years ago. Okay, so this is just to put things on perspective. I guess most of you are around 22 years old. Writing history is 10,000 years old, but life is 4,000 million years old. So that means that all the extant living beings we find today are here thanks to an uninterrupted chain of reproduction events that have been going on for 4,000 million years. All right. So that's, for me, is mind-boggling. So we are here because there has been those reproduction events going on continuously for 4,000 million years. If you were to prune one branch 1 million years ago, maybe we wouldn't be here. Okay. So next time you step onto a little critter in a little beast, Remember that you might be exterminating an evolutionary lineage that has been going on for over 4,000 million years. Okay, so the problem with this conception is that you can become a little bit obsessed and you start to see evolutionary relationships in everything, all right, in everything that relates you. So now I have a little exercise for you. I will give you just 1 minute. I would like for you to look at those toppings that you find in a pizza and sort them out based on how closely related they are to you. Okay, so I want to find your personal relationships with the pizza toppings. Okay, and to help you, here you have the phylogenetic position, the evolutionary position of all those items in the pizza. Okay, I will give you just 1 minute, and we'll check back with you to see if you came up with the right solution. All right, let's see, how did you do? So basically, pepperoni, you share a last common ancestor with pepperoni. Pepperoni is made out of pork, of pigs, 180 million years ago. And you have tuna, which is a fish, which is a vertebrate, and you share a last common ancestor 500 million years ago. Shrimps are arthropods, and we share a last common ancestor with them 550 million years ago. And then we have mushrooms, followed by olives. I guess most of you know that we are more closely related to mushrooms than mushrooms are related to plants. Many people think that fungi are plants, but no, actually they are more closely related to animals than to plants. Okay, I hope you did well. And now to finish, I would like to show you some examples of what can we do with that, okay? Well, one obvious application of those evolutionary trees is to classify life, okay? So we can use those trees to generate a classification of live beings. And this includes everything, including viruses and bacteria, okay? And we use those tools a lot to classify bacteria. But one of the uses that, one of the applications that excites me the most is to reconstruct the past, okay? So we can apply something called evolutionary thinking. So for example, let's say that this is the evolutionary tree of a group of organisms, and you know that those ones in green share a specific feature. Applying evolutionary thinking, you can estimate, you can infer that this feature emerged in their last common ancestor. Okay? And we can apply this to anything that is biological. Okay, as far as we know, the features are homologous, are equivalent. We can apply it to everything. So going back to vertebrates, the evolution of vertebrates, we compare all the features of vertebrates. We can infer that, based on the phylogeny, that the milk emerged in the first mammal, the last common ancestor of all mammals. The amnion, this layer in the placenta, emerged in the last common ancestor of turtles and leopards. Then the four walking legs emerged in the last common ancestor of amphibians, reptiles, and mammals, and so on. So we can reconstruct and date the origin of all those features, which is essential to understand how animals evolve. Okay, so and that's very cool because phylogenies are the closest thing to travel back in time. Okay, they are comparative biology is a very powerful tool. Okay, for the sake of time, I will explain this very quickly. So if you want to have a full explanation, you have to pay the fees or get a scholarship and come here to Bristol, and I will give you the full explanation. But basically, the way in which we reconstruct those trees is by comparing those features across animals. We can do it using anatomical traits like the hair, the milk that I was talking about. But nowadays, we do this using molecular sequences. We use genes, okay? We compare the genes between different animals, and when we use them to reconstruct the history of those animals. So what can we do with those tools? Very cool things. Probably many of you have heard this traditional classification of live beings into bacteria, plants, animals, protists, fungi. Okay, this became totally obsolete 44 years ago now. Okay, so the scientist Carl Woese, he used molecular data to reconstruct the evolution of all life. That's amazing. I mean, there is probably no most important question, but it's one of the most important questions in biology is how all of life have emerged and how it's structured and how all life beings are related. Okay, and for example, here in Bristol, we have people like Tom Williams, who are very, they are top-notch, they are worldwide experts on this topic, the origin of life and the structure of the tree of life. Sorry. Just 2 weeks ago, he published a paper on Science discussing how the first cellular being looked like. So how amazing is that? No, the metabolisms, the structures, etc. That's amazing. And there are many tools you can use. I don't have time now, but you can use these tools later. If you Google, for example, for OneZoom, is this page here in which you have all the tree of life, and you just can travel around. For example, you can look here for humans. Hope you don't have vertigo. And it takes you from the root of the tree of life to humans. Very awesome. And if you do this in a smartphone, it's even cooler because it's touch sensitive. What are plants from here? So you can zoom out. You travel all the way to plants, for example, there. And then you can try to find the common relationships. So you have human light and you have plants on the other, and then this will tell you that the last common ancestor was here in eukaryotes. Okay, so there are many tools that we use to teach those things, and this other one, life map, that you can also use. So just Google them, OneZoom or life map, and spend. If this was a real lecture, I would allow you to play with it for a little bit. It's very cool. What other things we can do? Well, as I say, we can apply tree thinking to reconstruct the past. So in my research, I have used this to reconstruct which genes were present in the very first animal, okay? And that's amazing because it's telling us a lot about the biology of animals, which genes are shared by all animals, which genes are never lost, which genes are only found in animals. And we have done this not only with animals, but also with plants. And we have discovered many genes that nobody has seen before that are essential to understand, for example, how land plants became adapted to life on the terrestrial ecosystem. But as I said at the beginning, those tools are very transversal. I mean, you can use those tools to study anything that has genes and that has evolved. And that applies to all life beings, everything. So for example, we have an ongoing project in which we are comparing genomes of parasites, animals that are parasites, to try to find genes only found on parasites. And with the idea of using those genes as candidates to drug targets, to develop drug targets. So we're trying to find genes in parasites, only found in parasites, that we can use to develop medicines to fight parasitic diseases. Or we are comparing the genes of plants, the genomes of plants that are very adapted to drought, that they survive very well when there is no water. And we're using evolution to find those genes and now try to engineer those genes into crops, okay, to increase food security. Another researcher in our school, Gary Barker, for example, also used these kind of tools to study the coronavirus. Phylogenies are essential to understand viral diversity and evolution. And for example, they were very important to trace, well, they are very important to trace how the different variants have emerged and how the virus have dispersed over the world. Or for example, we use those phylogenies to reconstruct the hosts of the viruses. All this debate that there was about the host before the spillover being a bat, being a pangolin, was based on phylogenetics. Okay. And for the sake of time, I'm going to stop here, but basically you can do phylogenies of everything, languages, Pokemon. It's very, very interesting. So if you are more interested, if you're interested in learning more about phylogenetics, I would suggest you to check these couple of books, Richard Dawkins, The Ancestor's Tale, or The Tangled Tree by David Quammen. And with that, I will stop talking here.
That's great, Jordi. Many, many thanks. I noticed we've got a question in the Q&A there from Adrian Munoz Bucio.
Yeah.
Is there any gene to construct a phylogenetic tree from bacteria to humans? For example, the one that encodes some unit of the RNA polymerase semi-conservative gene. Hope I've got that right.
Yeah, you got it perfectly. Adrian, that's a great question. And the answer is yes. There are actually not many, but a few that are shared by bacteria, archaeas, and all eukaryotes, okay? And you got a perfect example because the genes that are more conserved are usually involved either in what we call information processing. So this is everything that has to do with DNA replication, transcription, and translation, or they're involved in metabolic functions, okay? Very basic metabolic functions that are found in all living beings. So yeah, there are such genes, and those are the kind of genes that, for example, my colleague, Tom Williams, uses to reconstruct the tree of life.
Thank you, Jordi. Thanks for the question there as well. Well, we've got another question appearing now, starting to get people in. That's good. Madeleine, how genomics help us determine when a species ends and another one begins?
Oh, that's another great question. That's a tricky one, all right? So I'm going to talk mostly about animals and plants. So I'm cheating because bacteria and single-cell eukaryotes are much more complex. But basically, there is a bit of a debate of how to do this. So one option is to compare the same gene across different animals and plants. Many people use mitochondrial genes to do this. And then they set up a boundary. If two animals are more different in the gene than 97% of the nucleotides, then we say that those are two different species, okay? The problem with that is that animals or plants are very diverse, and in some groups, the boundary should be 90%, and in other groups, it should be 99%, so it's a little bit hard. However, with genomics, now we can do something very cool, that is to check if genes travel between one population or another. One of my favorite examples are Denisovans and Neanderthals. Okay, those are close relatives of humans, of ourselves. For a long time, there was a lot of debate between anthropologists if Neanderthals and humans had some genetic exchange, to put it in a polite way, all right? But we never knew. Now we can sequence the genome of Neanderthals, Denisovans, and humans, okay? We have genomes of humans that died 40,000 years ago. The technology is starting to be crazy. A couple of months ago, they sequenced a genome of a mammoth that died 1 million years ago. Anyway, you check the Neanderthal genomes and the human genomes, and then you can see that they share genes that are very close, that they are very similar, genes that are too similar to not having exchange between populations. So then you can say, okay, if these two populations, Neanderthals and modern humans, share genes, that means that there was genetic flow, okay? So we can use this to define a species.
Great. Thank you. Another question popping up here from Elder. Can you tell us about the use of the molecular clock to date splits in the phylogenetic tree?
That's a fantastic question. Those are all great questions. So I didn't have time to talk about all my colleagues. In the School of Biological Sciences, there are two big professors, Davide Pisani and Phil Donahue, and they work doing exactly that. So basically, long story short, we can compare genes to reconstruct those evolutionary trees, but now we can count, using those trees as framework, we can count how many differences are between those genes. No? Maybe between a human and a chimp, there are 2 mutations, but between a human and an elephant, there are 5, okay? Now we can check the fossil record, and if we know that chimps and humans diverged 5 million years ago, or humans and elephants diverged 180 million years ago, now we can correlate the number of changes of mutations with dates. And now we can date splits in the tree for which we don't have fossils. So yeah, we can. It's a very powerful tool and very cool that we can use to date those things, like the origin of life, for example. All those numbers I was giving you, like the 4,000 million years for the origin of life, those are results from Phil Donahue and Tom Williams here in the school. So yeah, that's very cool.
Thank you, Jordi. Another question from Diego this time, who fell in love with the topic. His question is there an evolutionary or genetic relationship between the parasites that you presented in your study with their hosts?
Yes, those are very fun, amazing questions. So it depends on the parasite and it depends on the host. So it depends on the pair parasite host, okay? Most of the times what you have is co-evolution, Okay, so you can do a phylogeny of parasites, flatworms, no? There are many like the Tenia I showed there, or you have the flukes like a schistosoma, or maybe you can check roundworms, no? Like the filariasis, trichinosis, all those kind of diseases. So you can do a phylogeny of the parasites and a phylogeny of the hosts. Many times you can see that there is a correlation. Okay, there are no cases, or I'm not aware of many cases in which a parasite is very closely related to the host. Okay, mostly because parasites are parasites, and then they are not very good hosts because they are missing the things that they need to become a parasite. Okay, so yeah.
Very good, thank you. And Fernanda, also loving the talk. I saw in one of your phylogenetic trees you used an outgroup. How do you determine which species to use? Does it have to be completely unrelated? why the other trees didn't have them?
Thank you, Fernanda. That's another great question. Okay, the outgroups. When you define an outgroup, it's an experimental decision. Okay, it's not based on the tree. So, for example, if I am looking at vertebrates, I need to define an external group for comparison that will allow me to infer the tree. Okay, that in the case of vertebrates will be a close related invertebrate. So, but this is, as I said, this is an experimental decision. Sometimes you don't need an outgroup. Well, you need an outgroup when you want to polarize the tree, when you want to give it direction, when you want to say this is the oldest part of the tree, the root, and this is the newest part of the tree. But sometimes you don't need to do that. So, for example, when I study gene family evolution, I reconstruct the evolution of genes rather than the evolution of a species. I don't need an outgroup because the only thing I want to do is to polarize, to cluster together, to group together different groups of genes, all right? But in other cases, like the coronavirus, it's very important to have a temporal framework. So yeah, usually an outgroup is something you decide, and it's something that is close related to your in-group, to the one you want to study, but you have to be sure it's not inside the in-group.
Thank you. We have another question, this time in the comments, again from Madeline. Not sure it's the same Madeline. Where would you place the tree of life, or you wouldn't place it over there? Why or why not?
Okay, that's a fantastic question. And it's another source of debate. If you check, if you Google my name and virus, I have an article trying to explain that. So there are two competing hypotheses about where viruses come from. Hypothesis number one says that they are degenerate cells. So in the past, ancestors of viruses were full cells with membrane and everything, but they lost it. The second explanation, the second hypothesis, is that they are rogue genetic elements. So there is this group of genetic elements called transposable elements, or jumping genes, or transposons, that basically they copy-paste themselves or cut and paste themselves inside your genome. 40% of our genome is made up of those transposable elements. So there is the theory that some of those elements were able to escape cells and become viruses. Okay, most of the evidence supports the second hypothesis, that viruses come from they are from transposable elements. Yeah. So I wouldn't place them on the tree of life, or I would place them so. Animal viruses will have evolved from animal genomes or from transposable elements in animal genomes. Yeah.
Thank you. Question from Eric. Oh, they're flying in now. What is the relation between virus and evolution or species?
I will try to reply very fast to the questions because I'm concerned that Sergio, the student, is here. But I love those questions, so I appreciate that you enjoy the lecture. Relationship between viruses and the evolution of a species. Very important. Mostly because viruses, there is this hypothesis of the red queen, in which there are species that predate on other species, and then one species evolves to escape the first species, and then the first species evolves back to chase the second species. So viruses are big driving forces in evolution because they keep making our genomes to evolve fast to overcome what do they do. Going back to the example of the Neanderthals, many of our immune system genes come from Neanderthals. So most of our immune system genes come from Neanderthals, and they are there because of viral diseases that existed 100,000 years ago. So yeah, very important.
Brilliant, thank you. Ugo asks, which phylogenetic tree represents the tree of life or a specific case of study of a taxon rooted or not rooted?
Well, again, it depends on what type of question you are making to the tree. Okay, even if we do those analyses in a computer, they are no different than experiments in the lab. And then you have to define parameters. You have to define the conditions to answer a specific question. So depending on the taxon, if I am looking, for example, at the evolution of genes, I explained before, then I might use an unrooted tree. But if I'm trying to reconstruct the temporal order of events in the evolution of animals or doing molecular dating, then I use a rooted tree.
Thank you. And Diego is back with another question. If you consider that genes and variation have a greater weight than natural selection for the evolution of organisms.
So actually, I think the two of them are related. I didn't explain natural selection because I wanted to focus on phylogeny, but I teach natural selection and all that in the school. So basically, natural selection needs variation in genes to work. Okay, it doesn't matter how big is the variation of genes. If there is a genetic variant that is bad, it will be removed by natural selection, and if it's good, it will be retained by natural selection. And we are seeing this a lot, unfortunately, with the variants of the coronavirus, right? That's what's happening with the virus. It's a fantastic example. I know that it's a very sad situation. It's very tragic, but it's a fantastic example of evolution because you can see how all those variants emerge. They provide to the virus with an advantage to become more infectious or to spread more, and then they get selected. Okay, and remember, sometimes I hate because in the media they say the virus evolves to avoid the immune system or to avoid the vaccine. So that means that the virus has some kind of will to evolve or some intelligence. No, it's not like that. You have a million different viruses. One by chance is lucky and has a mutation that allows it to survive or reproduce better, and then given enough time, it will become more abundant in the population. Okay, so yeah, I think you need variation, genetic variation, to have natural selection. So, and it cannot overcome natural selection.
Thank you. And Oscar asks, humans can evolve to be immune to animal-borne parasites? Totally.
Yeah, totally. Yeah. Many examples like the flu, the seasonal flu we suffer. I'm not a specialist now, so don't quote me on what I'm going to say now. But many of the flu variants we suffer during winter, many of them originally come from other animals. They come from bats, they come from birds. Actually, there was a problem with the chicken flu 10, 15 years ago in Europe. That was flu that was able to jump between animals. But we can evolve it. But evolution means that some of us will die, and the ones among us with the genes to survive the virus will pass it to the next generation. Okay, so I don't know. I guess it's good for the species in the long term, but it doesn't mean that you are going to evolve resistance.
Well, thank you. Adrian asks, is there any information about the first viruses or how many mega million passed from the first viruses? Could be bacteriophages like the first viruses?
That's amazing. So basically, if the second hypothesis I explained, the origin from transposable elements, is true, that means that viruses have evolved multiple times during evolution. Okay, so I don't know how many times, I have no idea, but that means that within bacteria you have multiple phases evolving independently over time, and the same with eukaryotes and so on. We don't have, for obvious reasons, we don't have fossils for viruses, so it's very hard to date when they did emerge. However, we can check, that some viruses, they can leave a fingerprint in the genome in bacteria or eukaryotes. Okay, they leave a molecular fossil in the genome of organisms. So that means that we can compare the genomes of bacteria or eukaryotes, and we can say, okay, this virus was already present when the first vertebrate evolved, because this virus has been integrated in the same place in all the vertebrates that we check nowadays. Okay, I don't know much about bacteria, but I know, for example, about many cases of viruses integrated in the genomes of vertebrates. Okay, and many surprising findings, like most of the viruses that impact human health, they come from viruses that originally were living in seawater, marine viruses. So So yeah, we can learn a lot by comparing the genomes of different organisms.
Fantastic. Thank you, Jordi, for answering all those questions, and thanks to those who asked the questions. Yeah, very good questions coming in there. So thanks very much. I wanted to pass on quickly because Sergio Hidalgo is also joining us. Sergio, many thanks. I can see you on screen now. Thanks very much for joining us. Sergio is a former PhD student from the faculty. Sergio, many thanks for joining us. We invite you along just maybe to share your own experiences of studying your research in Bristol, and maybe what you've been doing since then, and yeah, just give us a bit of a flavour of what you learned and studied when you were in Bristol. Thank you very much for joining us.
Thank you very much for the invitation. Yeah, can I share screen?
You should be able to. Is it allowed to? Yes, there you go.
Yeah, perfect. Okay, thank you, David. Yeah, as David mentioned, I'm a former student of the University of Bristol. My journey through grad school was kind of difficult, messy, because it wasn't just in one university, but it was really fun. So just to talk to you a little bit about what was happening in the meantime, I started in Chile, actually, where I'm from, in the Pontificia Universidad Católica de Chile, working with Dr. Jorge Campuzano. I did my undergrad studies with him and the same university, and then I started to do my PhD there. And then it just came out the opportunity to move to Bristol to continue my PhD as a part of a joint degree. So basically, I jumped into this opportunity and I went into Bristol, and I moved to this amazing city. If you have any questions, we can discuss about it, but the city is amazing. I arrived, the university is beautiful, and I then started to work with Dr. James Hodge in the biomedical sciences building. This is one of the many other activities that we did in the lab during the time. This is James over here. And the good thing about Bristol and is about my experience there is that it allowed me to continue part of the work that I was doing in Jorge's lab, working with the fruit fly, basically. And I did my PhD thesis in both labs, partially working with Jorge and partially working with James, and using this fly, this animal, to model schizophrenia symptoms, basically. So what we did in this case was to take the fly, try to characterize genes that were hit on GWAS studies, and then try to find molecular and behavioral markers of schizophrenia. Sounds weird, schizophrenia in flies, and actually it is. What we did basically was to range from behavioral tasks. We did electrophysiology, we did calcium imaging, and a lot of confocal imaging. That was one of the amazing things of moving to Bristol. One of the things I enjoyed the most was the imaging facility, as Alec mentioned at some point. And I was working here at the Physiology, Pharmacology and Neuroscience PhD program. And one of the good things that for me was actually to be able to contribute to a lot of publications, and I learned a lot, actually, new techniques, and basically by moving to Bristol, just a little bit of briefing here. We were able to publish a couple of papers in my brief time there. It actually was really productive from 2018 to 2020 that I was in Bristol. We managed to publish this study showing calcium channel blockers and Alzheimer's disease using the fly, neonicotinoids in bees and flies in another publication. And this is actually part of my PhD thesis. The two publications came out showing the work that I will that I was doing during my PhD thesis per se. So this was a great opportunity to work with new people and to basically use all the facilities and possibilities that the university has. And actually that opened the doors for me to look forward to opportunities. Actually, right now I'm in California. I'm in the University of California in Davis, in Dr. Joanna Cheer's lab. Dr. Joanna Cheer's lab work in circadian rhythms, actually, and I fall in love with circadian rhythms while working with James. So actually that shaped my entire career in which I'm doing now. That is basically understanding the molecular clock and its contribution to physiology. So the university actually opened a lot of paths for me, and this was one of them. I think that going through the university, obtaining the degree there, opened the possibility for me to go to these new environments and new opportunities. So yeah, that was pretty much it. What I need to tell you about my experience with the university and how it led me to what I'm now.
Many thanks, Sergio, for your brief presentation, for sharing with us your experience in Bristol and what you've been doing.
Sure.
And really interesting, and thanks very much for being with us and sharing that. We had a question during your presentation there. I must admit it caught my eye as well. The question is, how do you know that a fly is schizophrenic?
Yeah, so this was fun from the beginning, because when I actually came out with the project that I wanted to see schizophrenic flies, both Jorge and James were kind of reluctant at the beginning and asking the same question: how can you know if a fly is schizophrenic? And the question is, you cannot know. It's impossible to know if the flies are schizophrenic or not. What you do is to model the disease. That is slightly different, but basically we based our research on the genetic component of the disease. So basically schizophrenia is around 80% of genetics, and a lot of GWAS studies have been conducted, and we take advantage of these GWAS studies, and we pick those genes that were recently discovered as associated with the disorder, and try to find the homology of those genes in flies to try to modulate them, knocking down, overexpress them, to see what happens to the flies. And specifically, we'll look at behaviors that are usually modified patients with schizophrenia, for instance, sleep disturbances, and circadian locomotor disruptions, learning, memory, or cognitive problems, and all of those you can actually measure in this animal Drosophila by doing some behavioral tasks. So yeah, you don't know if the fly is schizophrenic. You are just basically taking a bunch of possible behaviors that can be linked to it. But yeah, I don't know if the flies are hearing voices or having any other. I mean, I cannot ask them.
Very good. Thanks very much for that answer, Sergio. Yeah, killed some curiosity there. So many thanks again, Sergio, for joining us and for sharing your experience. Elec, Jordi, many thanks again. I don't know if you'd like to offer some final words on the session. Oh, we've got another question popping up there. Anybody like to pop in? Just Diego again. Well, a question from I think it's for you again, Sergio. How did you measure the response to antipsychotics?
So basically, what we did in both works, they're already published in open access. So if you want to go and read them, please do. They're out actually this week. Yeah, basically what we did is to measure these behaviors like sleep, memory disturbances, circadian locomotor activity, and then we gave the flies that were mutant for those schizophrenia associated genes these antipsychotics like classical antipsychotics as haloperidol, and basically see if some of these components that were disrupted by mutating those genes, if they are recovered after giving them the drug. So basically the flies presented less locomotion, an increased locomotion during the nighttime. If you give them these antipsychotics, you can see how the locomotor activity goes back to normal. So this is one of the ways, and we did with a couple of other behaviors in the study. So yeah, basically trying to see if everything goes back to normal with the antipsychotics.
Brilliant. Many thanks, Sergio. Well, I think we're coming to the end of our session now. It's been a great session. Jordi, Alec, Jordi, I see you're back online. Any final comments you'd like to make before we close for today? Thank you again for your time and giving up a good portion of your evenings in Bristol. Any final comments, please?
I would just reinforce something that Alex said, and I think Sergio's research is a perfect example. Keep your minds open to everything in biology and biomedical sciences. It's very, very exciting. There are so many cool things you can do. Come here is a fantastic place to live and to research.
Thank you, Jordi.
Yeah, same here. So Bristol is a fantastic city. Many of us came here and remained here because it's a beautiful city, very collaborative, friendly environment. It's a great university. We genuinely like to work with our students. And if you are interested in any of the MSc programs, please come to the faculty website. You can find the full description of various units and research projects and so on. If you are interested in PhD or MSc by research, then don't be afraid to contact directly any of our academic staff. They are expecting this. So we receive these sort of inquiries on a regular basis. Do a little bit of research. Look at the website. Identify research areas which appeal to you, and send them an email. Introduce yourself briefly. You can ask questions about current research activities, and then you can start a dialogue which could lead to a MSc by research or PhD placement. We are also very happy to provide advice regarding fellowship applications if you are eligible for any of the schemes. If you have an idea how you would fund your PhD studentship, we can certainly help you to come up with a research project. So plenty of support is available here. Don't be afraid to use it. And if you have any questions, then feel free to contact David or Gary or international office, or you will find lots of email websites. If you have academic questions, then Jordi and I are also happy to guide you to the right direction, even if we can't answer it. So thank you all of you for joining us. It was a pleasure, and I learned a lot about evolution today.
Fantastic, thank you. Yes, well, we will be following up with all the people who were able to join us live today, and as I shared, we had many more registrants. Not everybody's been able to join us today. We had a great group of people with us today. We'll share your presentation. The session's been recorded. We'll share that with everybody, and hopefully we'll have lots of people coming back to us with questions and asking for more information. And myself, Gary, who's now been able to join us, will be happy to answer any admissions type questions, and Alec, Jordi, and other colleagues will be able to help with the academic side of things. So I think we can conclude today's session for now. Thank you again very much to Alec and Jordi in particular for giving up some of your evening today and for giving such stimulating talk. And thanks to everybody who's participated and been a part of the webinar today. I think we should try this again sometime in the future. So great to have everybody with us today. Thank you, and take care, everybody, and look forward to hearing from you and to supporting your applications and your requests for information from Bristol. Thanks to everybody. Have a nice evening. All the best.
Thank you very much. Nice to see you.
Bye for now. Take care.